Hey guys, so today we're going to be working on our second lab of the program, and it's our cancer detection lab. You should have gotten a kit like this from Carolina. It's their Hudson Alpha edition of the HNPCC lab. And in this kit, you're going to get a couple of things. This is your instruction manual. A lot of the information that I provided for you in the previous little video came from this manual, so it's got a lot of good stuff in there. I highly recommend you look through it. You're going to get your DNA samples, and this particular kit has enough samples for 8 lab groups. The premise of this lab is we are going to be tracking a cancer gene through a simulated family, and they provide you 4 copies of the family's DNA. So pretty much what they're looking for you to do is to take one copy of the family DNA and divide it up amongst 2 groups. If you have a class that is smaller, then you can give one full family to one group and you can have them run a double gel, which I'll show you how to do today. Other things that you'll get in the kit, you're going to get some agarose powder with whey boats and gloves so you can make your agarose gels. However, I'm going to show you a much simpler way to do that in a little bit. You are also going to get some gel electrophoresis buffer. The buffer that comes with this kit is a 20x concentrate. I'm going to use a 5x concentrate, but I will show you how you can dilute both. And then you're also going to get some stain that you can use to stain your gel in the end to visualize your DNA bands. Personally, I've never used this particular stain, even with this kit, and I'm going to show you a much more simpler way that we can do this in our own home and view our DNA results immediately, and I think you'll love it. So there's 2 different gel electrophoresis systems, and I'm going to focus on one today that might look a little bit unfamiliar to you, and that is this blue gel system from a company called HNPCC. And it's awesome. It's got many advantages to using it, both if we're going to do some at-home labs, if we have to go to e-learning in the fall, or just for you guys to do this right now through this program. But it also has some really cool advantages using in the classroom. One, if your kids are new to gel electrophoresis, or even if they're more experienced in the lab. And it also reduces a lot of the steps that tend to be bottlenecks when you're doing a lab like this with a class. So we'll talk about that as we go through the system. So you understand the components of each. There is this blue gel system that you get from HNPCC. Has a casting tray system, which is one of my favorite parts of this lab, and that will replace something that's more traditional that you might be currently using at your school. This kind of looks like what I have to use. I have a gel casting tray that looks like this, but mine's even a little bit more archaic. I have to put electrical tape on the sides, and that comes with a lot of problems. A lot of my gels leak when I pour them. So I love this little guy from HNPCC. But in addition, if you do get this from the Baxter Center of Science Education, it still works really well. I didn't have any problems with the gels leaking once I poured them. These little rubber stoppers pop on nice and easy. But I did happen to tear the gel when I took these rubber stoppers off. So anyways, this guy is awesome, and you'll see when I pour the gels in a little bit. This is going to be your gel electrophoresis chamber that comes with the blue gel system. Inside of it, you have this little buffer chamber, and then the base of it is going to provide the power that will hook up basically to the electrodes in the cap that will provide the current. So this gel electrophoresis chamber is going to replace this big tub here. And then the top part, you have these electrodes. Like I said, that's going to plug in nice and easy here, and we're going to have our current that's going to run through. So this orange cap is going to replace our more traditional cap that you'd have to place onto the gel electrophoresis chamber in a traditional system. One other major advantage of using the blue gel system is we will be able to visualize our DNA samples immediately. There's a light built in here, and as soon as we turn that on and we look through this filter, we will be able to see our DNA bands move in real time, and the kids can easily capture pictures, or you can capture pictures as the DNA bands progress down the gel. A few just random thoughts that I've had over the years when I've been doing different labs. One thing that I really like about this cancer detection lab, it's a great gel electrophoresis lab to start the year off with, because these samples are pre-digested samples. And so that means that basically the only thing that we have to do to run this lab is to make our gels and then make our buffer and and then pipette the samples from this kit directly into our gel. So there's no other sample prep that we need to do because they have already been digested with a restriction enzyme. So there's no restriction digest. This is not a PCR lab. In my classroom, some of those labs come in later in the year once the students have really just mastered the technique of gel electrophoresis. Another just general comment or thought and question that I've had over the years is, if I'm buying a consumable kit from a company, what systems are they compatible with? This system, like I said before, comes from a company called HNPCC, and you can run any consumable kit on this system. So if you get a kit from Carolina, if you get it from Biorad or Edvotech, all their samples will work with this system. Likewise, HNPCC has their own consumable kits, and those are compatible with any of the more traditional systems as well. So, you know, if I know a lot of us don't have this in their classrooms. I don't have this kit in my class, or this system in my classroom. I have to reserve it from the Baxter Center of Science Education. But I do have a more traditional system, and so I can still use some of those HNPCC consumable labs on my traditional system as well. But we're going to focus on this one today because I think this one will be new to you, and I guarantee that you're going to like a lot of the components of this blue gel system. All right. Okay, so we are going to make our buffer. The buffer that we're going to make is going to be used to make our agarose gel, and it will be used in the buffer chamber when it actually comes time to run our gel. There are two concentrates that you may have received. One of them is going to be the 20x TBE buffer that came in the Carolina kit that you got, and then the other one is going to be possibly a 5x concentration that the Baxter Center dropped off for you. Either one, doesn't matter which one you use. I diluted it with spring water that I just got from Jewel. Worked great. I've already tried one gel. The main thing is that you just don't want to use tap water. Now, I made up 500 milliliters of a 1X buffer for myself just so I could run a couple of gels and make sure that everything was working okay in my house. So if you want to make up a little bit extra and therefore you don't have to keep remaking buffer, I think it would be a good idea. If you're going to make 500 milliliters, that might be a lot. You can use the equation C1V1 equals C2V2 and make up any volume that you want. If you're going to make up 500 milliliters, that means that you're going to use, in the 5X concentration, you're going to use 100 milliliters of the concentrate and 400 milliliters of water. You should have gotten a graduated cylinder, so that will help you out, but you could always use measuring cups as well. You just need 4 parts water and 1 part of this concentrated buffer solution. If you're going to use the 20X, then that means that you need 19 parts water and 1 part of this 20X concentrate. If you make up 500 milliliters of buffer, then that is going to be 25 milliliters of this and 475 milliliters of water. So I previously already diluted mine. So I have some 1X TBE here, and believe it or not, I could not find any tape or Sharpie, anything that I could see on video in my house. So it is unlabeled. Next, what we're going to do is we're going to make our agarose gel solution. Now, I showed you previously that this agarose powder comes in the kit from Carolina. This would be great if I was at school. I do not have a scale in my house, but I do have these little agarose tabs, and these agarose tabs are amazing. They are 0.5 grams of agarose solution, and we are going to make the percentage of agarose solution that you want to make really depends on what lab that you're doing. This particular lab in Carolina calls for a 1.2% agarose solution, but I have used 1% gels many times with this lab, and it's worked great. If we're going to do that, that means that we need this 0.5 gram tablet is going to go in 50 milliliters of buffer. For once, I am actually going to try the 1.2 concentration, but if you want to use the 1% concentration, feel free, your lab's going to work. I just kind of want to see if I'm going to get some better separation with a higher percentage of agarose. So that being said, I think that works out to 42 milliliters of buffer. So I'll just measure that out here. And I do have a couple transfer pipettes, so I don't really have to go back and forth, but I'm not going to use them. All right, 42 milliliters, close enough. And all we're going to do now is take this tablet. Again, this is 0.5 grams of agarose, and we are going to dump it into this flask. And we're going to let it dissolve for about 4 minutes, and then I'm going to pop it in the microwave for 45 seconds, and we should see that this has completely dissolved and become transparent. So as this sits here, you'll start to see it dissolve, and if you're not really familiar with pouring gels, this will become very opaque, and you want to make sure after you heat it that it will become very, very transparent. So I'm just going to let this sit here for a couple minutes. Then I am going to go pop it in the microwave for 45 seconds, and I'll be right back with you. Okay, so I just melted that agarose tab for exactly 45 seconds in the microwave, and you can see it's completely transparent. So that's what you want to see. If it's opaque at all, then pop it in your microwave back for a couple more seconds. I did not get any boiling over. I know all microwaves are a little bit different, but 45 seconds and it did the job. Okay, so now comes one of my favorite parts, this casting tray. I love it. On the bottom, that's where they keep their combs. So we're going to pop these 2 combs out. Now, this particular Carolina lab that we're doing, I think there are 14 samples in total with the controls and all of the family members. So we are going to be able to use the larger teeth on the side of the comb, and we're going to make 2 rows. So one thing about this casting gel really quick, or casting tray, there is a certain way that the kids can put this in. So if you look on the sides here, there's 2 ridges on one side and there's 1 ridge on the other side. This can only fit in the casting tray in a very certain way, and therefore you'll see as we progress through all these steps, there's only one way that this casting tray can go into our gel electrophoresis chamber. So really, the kids cannot mess up the orientation of their gel in relationship to the positive and negative electrodes when we're actually setting up the gel electrophoresis, when we set up the power. So when you put these combs in, there's 2 little slots that they fit nicely into, and this white tray here is heat resistant, so it won't warp even if your agarose is really hot. So you don't technically have to wait for this to cool down, which is really nice. And we should not see any leaking with this. So this, I know it's very simple, but obviously I've been traumatized by my past with pouring gels. Okay. We're going to use this dye. It's called gel green, and this is what is going to allow us to view our DNA samples immediately once we've put them in our gel electrophoresis chamber of the blue gel system. Now, if you are going to run your gel on a more traditional system, you can still use this gel green. It's just in the more traditional chamber, you won't be able to visualize the DNA bands. You're going to have to still view your gel either over a UV light transilluminator or over a blue light to actually see your DNA bands. But this gel green is what eliminates the need for us to do any sort of staining, overnight staining, once our gel is done. This is a 10x concentration that comes in this little tube. So that means you basically are going to use 10 microliters per 100 milliliters of agarose solution. So again, that's 10 microliters per 100 milliliters of agarose solution. I have 42 milliliters in here, so I'm going to use 4.2 microliters. If you decided to make a 1% agarose solution, then you probably have 50 milliliters in here, and so then you would use 5 microliters of this gel green. So I'm going to just adjust my pipette here and add in the 4.2 microliters. You can add this even if your agarose is still piping hot. So we'll get this gel green in there. Sorry, these gloves are really big. Okay, and you will see that this agarose is going to turn slightly orange once we give it a little stir here. So it's still very transparent, but now it has a little bit of an orange tint to it. And all we're going to do is we're going to, and I'm a little bit of a wuss, so I'm going to use this little pot holder here that I think my niece made for me at my wedding. All right, so we're going to pour this in here nice and slow, and we're just going to go about halfway up the combs. If you feel like your kids are a little bit less experienced, you can pour the gel up a little bit higher. But that's it, folks. There's going to be no leaking and there's going to be no problems, and this is going to solidify pretty quickly. You might ask why I'm not pouring a second gel, because I probably do have plenty of agarose in here to do that. I don't have any other combs, and I do want to make sure that you can see all of your DNA samples on one gel when we run our system, so that is why I put 2 of the combs on one gel and not one in each separate one. All right, and there's pouring your gels. We'll wait for this to solidify, and then we will start loading our samples. All right, and we're back, and our gel has solidified for us. So if you wanted to give it a little touch, make sure that it's completely solid. Don't poke it. We don't want to get any holes in there. We're going to remove our combs now, so we're just going to give each a little bit of a wiggle and a tug here. I don't want to pull this tray out quite yet. And there we go. Okay. All right, so we're going to take this gel out, and you can see we have this nice, beautiful gel here. Didn't leak at all. I know I talked earlier about my experience pouring gels. And great wells. And so one thing that I really like about this blue gel system that I mentioned before, this gel casting tray has 2 ridges on this side, and it has 1 ridge on this side. When we put that into our buffer chamber, that means that this gel casting tray will only fit in this orientation. So our wells are going to be up at the top here, and that's where our negative electrode is going to be. It is impossible for the kids to put the gel in this way, and it is also impossible for them to put this buffer chamber in this way, even if they oriented the gel properly in the buffer chamber. So this is foolproof for our kids, which is really nice. With my older students, sometimes I kind of like the fact in the more traditional system, it puts the pressure on them to really be cognizant of what they're doing. Sorry, I'm making this harder than it looks. Really cognizant of what they're doing. So this, in a way, I would have to emphasize to my kids to really pay attention to what orientation they're putting the gel in. We're going to add our buffer here. This is the same buffer that I used to make the agarose gel, and you can see this chamber super tiny, so we don't need to use a lot. And we are ready to start loading our samples. One thing that you're going to see when you get this kit, depending maybe on how old it is, some of the samples will have started to have evaporated, and you're not going to have a lot of fluid in there. There is supposed to be 20 microliters in the samples, and I tried to flick these down using like the human centrifuge way, and it didn't work very well. So what I'm going to do is add just a little bit of buffer to try get some of this down to the bottom, probably about 20 microliters of buffer that I just loaded into this tray, and then we will start loading our samples. All right, so I now added about 20 microliters of buffer to each sample just because they were so evaporated. Technically, we only need to add about 5 microliters per well, but I'm going to go ahead and set my pipette to 20 and just try load as much as I possibly can. I think I poured my gel with wells deep enough so I can get probably quite a bit of sample into each well volume-wise. So I'm going to go ahead and just load our samples here. The order of samples can be found in the Carolina manual, so I'm just going to follow that. They have a gel 1 and a gel 2. And I'm just going to use a new tip with each sample and try get as much put in each well as possible. Okay, we finished loading our gel. All we have to do now, and this is one of the huge advantages of the blue gel system, we're just going to pop on this top that has our electrodes. The negative side is going to go over here by the negative. There is only one way that this cap can fit on, so the kids cannot put it in the wrong orientation. We're going to pop that on, and then we just need to plug the power source into an outlet, and that's it. What's very, very cool about this is the kids can then now start checking their gel running in real time throughout the rest of the process. So all we have to do is hit this power button and the green light's going to go on and we'll have power. This light here is for a blue light, and if we just hit that blue light, that's going to illuminate our samples, and the kids can put this little box, which is like a little dark room, and pop it over the top and put their cell phone right here and snap some pictures. So this is going to be the end of the video, but I will definitely include some pictures of what I'm seeing as the DNA bands progress down the gel.